This morning I have the great pleasure to introduce Mazen Kheirbek, one of our newest faculty members, who's among the vanguard of young scientists who are who are figuring out how brain circuits control behaviors that are highly relevant to psychiatry, in his case, anxiety and mood control. And he'll be telling you about the role of various parts of the hippocampus in these processes. He has a fantastic CV at his young age, starting off at WashU, then I think University of Chicago and Columbia, where he did seminal work with Renee Henn in defining the roles of different parts of the hippocampus in the control of affective behaviors in mice. He became an assistant professor at Columbia until we were able to steal him. And I think it was a great recruit by Matt to bring Mazen into the mix of our young scientists in our department. He already has 2 R01s, so he needs no more help from his mentors like Will. He's won several awards, and his publication record is stellar. I think I'm on the promotions committee. Maybe we'll just go full professor next year and forget the rest. No, I can't do that. But so with that, I'd like to introduce amazing Mazen Kheirbek.
All right, thank you so much, John, and it's just a real pleasure to be able to come down here, or come up here to pronounce this, and share some of the work. So I am at Mission Bay, and so I moved here, like John mentioned, about 1.5 years ago. And so today I'm going to tell you a little bit about kind of some of the work that we've been doing, trying to understand kind of the contribution of different parts of the hippocampus, different circuits of the hippocampus, to different kinds of affective behavior. So just a brief kind of very, very general background of how kind of we think about the hippocampus. So we kind of think of the hippocampus at the interface of cognition and mood. And so there's this very kind of classical role of the hippocampus, if you read any neuroscience textbook, in kind of decorative memory, spatial memory, and contextual memory. So more kind of cognitive function. And so, so just a few years ago, the Nobel Prize was won by the Mosers for discovery of place cells in the hippocampus, which is thought to be important for kind of spatial navigation. And then we also know that the hippocampus is involved in different kinds of memories for facts and events and linking these things together. However, kind of there's this other side of the hippocampus, which is known to be involved in kind of more affective states, such as an emotional behavior and anxiety and different kind of stress responses. And so if we kind of look at kind of just a very, very general view of kind of some of the evidence for these, as I mentioned, kind of lesion studies with the classic patient HM who could not form new memories, had basically loss of his hippocampus, the place cells, the discovery of long-term potentiation, which is activity-dependent changes between neurons, is thought to be a neural substrate for memory, and in rodents that was first discovered at the dentate gyrus of the hippocampus. However, and so kind of the role of the hippocampus in emotional behavior and anxiety comes from a number of different avenues of study. So for example, we know that the hippocampus makes direct connections to different fear and stress circuits and can directly modulate the mounting of stress responses to different fearful stimuli. In addition, we know that stress can modulate hippocampal volume, and in a number of different mood disorders and anxiety disorders, the hippocampus is actually one of the first places affected. And then finally, one of the topics we've been very interested in is adult neurogenesis, which is the birth of new neurons from stem cells in the adult brain. And one of the few places in the brain, at least in the human brain, one of the only places this has been documented, is in the hippocampus, in the dentate gyrus. And so this is known, at least from the rodent work, to be tightly under control of the emotional state of the animal. And so just to give kind of just a few highlights here, so we know from this in the human literature, from this study from Gilbertson and colleagues, that basically that the hippocampus volume is very tightly correlated with the severity of symptoms in post-traumatic stress. So for example, this is a twin study where what they looked at is the actual the severity of the symptom of a combat exposed twin to the actual size of the hippocampal volume of the non-combat exposed twin. And what they find is that basically there's this correlation correlation with would indicate that actually a smaller hippocampus may actually be a risk factor for development of the disorder. In addition, kind of classic work from Yvette Chalene has shown that basically that the size of the hippocampus correlates with the time depressed in major depressive disorder, with the smaller hippocampus with a longer time in depression, depressed individuals. And then in kind of classic rodent studies from Bruce McEwen's lab, it's been shown that chronic stress can actually cause a shrinkage of hippocampal neurons with their dendrites, leading to dendritic atrophy, which may be important for these kinds of changes in hippocampal volume in response to kind of these disorders. So in addition to kind of changing the actual size of neurons and their dendritic architecture, another kind of interesting finding in the field has been that stress and enrichment, basically emotional states, can have very different effects on adult neurogenesis in the hippocampus. So this is a rodent study here, and what we're looking at here is the mouse dentate gyrus, which is the primary input point of the hippocampus. In blue here are the cell bodies of the dentate gyrus, and in green here is a label for adult-generated neurons. So this is labeling a protein called double cortin, which is expressed in cells that are about 1 month old and younger. And what you can see here under normal conditions, when the mice kind of just live in a normal cage in a mouse colony, what you see is you have these kind of young neurons that line what's called the subgranular zone of the dentate gyrus. So now if you stress the animal, and here the stress that's given is social isolation, what you see is there's actually a reduction in the number of new neurons that are born within the dentate gyrus. And in the opposite situation here, where you enrich animals, basically giving them a bunch of toys, a bunch of running wheels so that they can exercise, what you see is you see a growth in the number of new neurons in the dentate gyrus. So this phenomenon has been shown many times and with many different kind of manipulations of stress and enrichment. However, the actual function of these new neurons has yet to be really kind of elucidated and shown. And so that's kind of one of the main focuses of my lab, and I'll talk a little bit about kind of some of the headway that we've made towards understanding this. So another kind of way that people have been thinking about the role of the hippocampus in both cognitive function and mood is that it's actually functionally heterogeneous along its dorsal ventral axis. So it's been thought that the dorsal hippocampus actually may subserve these functions of kind of more cognitive behaviors such as decorative memory and spatial memory, whereas the ventral hippocampus is actually the area that's involved in kind of these mood and anxiety related functions. And so if we zoom in a little bit and kind of look at this circuitry, if we now look at kind of divide up the hippocampus into its dorsal pole and its ventral pole, so this would be anterior and posterior in a human, what you see is if you look at the connectivity of the hippocampus, so this is not super important right now, but this is basically the classic circuit of the hippocampus with the dentate gyrus, CA3 and CA1, and the subiculum and the output. What you see is that outputs from the dorsal hippocampus tend to send projections mostly to areas involved in context encoding and discrimination. Whereas the ventral hippocampus, if you look at the outputs from the ventral hippocampus, these tend to be areas involved in more anxiety and emotional responses, such as the hypothalamus, the amygdala, and the prefrontal cortex. And so what we're interested in understanding is basically how do local circuits within the hippocampus influence output of the hippocampus to control these specific behaviors. And so what we're focused on is kind of two main topics. First, the local circuits at the input of the hippocampus, and how these contribute to kind of the function of the hippocampus and behavior, and how this translates to long-range outputs of the hippocampus. And so what I'm going to tell you about today is kind of two stories. One I'm going to touch on just briefly because it's already been published, and one that's a newer story that we're excited about, which is related to first the role of the contribution of adult neurogenesis to behavior, and specifically to the generalization of fear memories. And then what I'm going to talk a little bit about is kind of the long-range circuits and the outputs of the hippocampus to these downstream areas to control specific anxiety-related behaviors. So I'll start with the adult neurogenesis, and as I mentioned, I'm just going to touch on this briefly since we've already published a lot of this work. So just to step back a little bit and why we think this is exciting. So adult hippocampal neurogenesis in the rodent, it's believed that about 3,000 new cells are born per day within the dentate gyrus of the hippocampus. And so here again is one of these pictures of a dentate gyrus, and now what we've done is we've labeled the stem cells and their progeny red. what we can see is that they basically line the subgranular zone, and they basically are born from stem cells, and they integrate into the local circuit. As I mentioned earlier, their survival is really dependent on the environment. So stress can reduce levels of neurogenesis in the hippocampus, while enrichment tends to increase levels of neurogenesis. Experimental studies in rodents have shown that ablation of neurogenesis actually impairs some forms of learning, some stress responses, and antidepressant efficacy. So for example, my previous mentor, Rene Hen, one of his major findings of his laboratory has shown that if you ablate neurogenesis in rodents, you lose some of the behavioral effects to SSRIs, such as fluoxetine. However, all of these studies tend to be these kinds of ablation studies where you basically remove all the cells from the circuit. But actually little is known about the online encoding properties of these new neurons in the hippocampus. So if we zoom in a little bit here and we kind of look at this process of adult neurogenesis, it's this really kind of unique form of plasticity in the adult brain. So it's basically this process takes about 2 months until cells kind of become mature neurons in the adult brain. So basically you start with these nestin type 1 neural precursor cells that give rise to type 2 transit amplifying cells, which basically divide very rapidly and give rise to the new dentate granule cells in the hippocampus. What's interesting about this process is that they basically go through a critical period of plasticity during their development, where these young neurons actually are very different from their mature counterparts. So what they basically show enhanced plasticity, and they may modulate local circuits very differently than their mature counterparts. So we know that they show a lower induction threshold for long-term potentiation if you look at slice experiments. In addition, they express receptors on their cell bodies that kind of confer this increased plasticity. For example, NMDA receptors that are highly plastic on their actual synapses. And so much of what we kind of knew about what these new neurons do came from in vitro experiments, where we basically look at a slice and we ask, how are these cells different from the mature granule cells? So what we wanted to ask is actually in the live brain, in the adult mouse that's behaving, what do these cells do? And so what the open questions that we've been asking is, how do adult-generated neurons contribute to memory encoding? And the second question that we've been asking is, how do adult-generated neurons actually behave and function in vivo? And so I'm just going to touch on a few pieces of data since we recently published this work. But what we've been using is optogenetic modulation. So this is the expression of light-activated proteins on cells in order to silence their activity acutely, and so we can determine exactly how they play a role in behavior. So now you've seen this a few times now, so this is again the dentate gyrus now of a mouse, but now what we've done is expressed a light-sensitive optogenetic actuator on the cell membrane of these cells. What we're using is ARCH, which is a light-activated proton pump, where if we shine light, we can actually silence the cells very acutely and reversibly. And so this opens up, the ability to do this opens up the possibility that we can now turn off these neurons in very specific patterns and ask how they're actually contributing to the behavior. And so as a first test, what we wanted to know is, are these adult-born neurons important for the encoding of a fear memory? So do these cells come online when the animal is required to learn that a context is fearful? And so in order to do that, we use a contextual fear conditioning test, which is a very common test in the rodent literature, where the animal goes into a context and receives a single foot shock. As a consequence of getting this foot shock, 24 hours later when they're tested for freezing to this context as a measure of their ability to form the fear memory. So now that we have control over these new neurons, what we can do is we can actually turn them off when the animal is learning that the context is fearful, and ask whether or not these cells are actually coming online when the animal actually needs to learn something. And what we find here is, if you look at the freezing of these animals, when we compare animals to control mice that don't express the opsin, what you see is you get a reduction in freezing when the animal basically is tested 24 hours later with the light off. So this would indicate that these new neurons are actually coming online when the animal has to learn that this context is actually fearful. So it suggests that they are actually doing something in the adult brain, and they are contributing to memory encoding. So now that we kind of set this up and we figured out that, okay, so these cells are important for actually learning about different contexts, what we wanted to do is kind of test a hypothesis that has been set forth in the field about what these new neurons are actually doing. And so a number of labs have argued that these new neurons are important for this process called pattern separation or fear discrimination. So the idea being is that if you take a rodent and you train them to discriminate between two very similar contexts, so the animal is put into a box and shocked, and then asked to discriminate between the box that they originally were shocked in versus a novel, safe context. Can they discriminate between safety versus danger? And you train them for multiple days, and what normal rodents will do is that originally they'll generalize their fear to the two boxes because they can't figure out the difference between them. And then eventually they learn to discriminate between these two contexts. So previous work has shown that if you ablate adult neurogenesis, so this is work from both Renee Hen's lab as well as Susumu Tonegawa's lab, has shown that basically if you ablate these cells, these animals have a very hard time discriminating between these two contexts. However, in this situation, we've ablated these neurons throughout the entire task. And so we actually don't know when these new neurons are actually coming online when the animal has to learn this task. And so we hypothesized from the previous experiment that perhaps these new neurons are important for learning new information. And so we hypothesized that if we silence them only when the animal has to learn that there's a new safe context that's different from the original fearful context, the animals would have a hard time doing this. And so now what we did is we turned back to our original optogenetic manipulation, except now only turned off the cells when the animal has to go into the safe context and discriminate that from when the animal has to go into the fearful context. And asked, can the animal learn this if we always silence the cells when the animal goes into the safe context? And what we found is that they cannot. And so even after 9 days of testing, silencing these cells only when the animal goes into a novel, safe context, the animals can't really discriminate between the two contexts. If we do this silencing when in the same context that they were originally conditioned in, we see no difference. And control mice can learn it just fine. So this would indicate that these adult-born granule cells are important for encoding novel information. And so this is important for fear memory encoding because this is important for the animal to be able to distinguish a representation that's very similar to one that was already fearful. So then the question became, how do these adult-generated neurons behave in vivo? And so in order to do this, what we use is now a method to be able to monitor the activity of these neurons in awake-behaving animals. And so the way that we do this is to express a calcium indicator. So this is a genetically encoded calcium indicator called GCAMP6. So basically this just changes its fluorescence whenever the cell becomes active. So now by labeling all the neurons in the dentate gyrus with GCAMP6 and using the same genetic trick that we used before to label adult-born granule cells, except now we label them red, what we can do is we can put the animal under a microscope and actually image the population activity of dentate gyrus granule cells in awake behaving animals. And so here what you have is a coronal section again of the dentate gyrus, very similar to what we've seen before, but now what we've done here is label all the young neurons red, and then everybody with the calcium indicator. So now by imaging above this area, we can look and find all the red cells and the non-red cells and image the population activity of mature granule cells versus immature granule cells. And so now what we can do is we put the animal under this, and so they're under a head fixation where they run on a treadmill that has multiple different cues. And so we can ask, how does the animal encode these cues in its environment as it runs along the treadmill, and then now look through the 2-photon microscope at the population of dentate granule cells. So here what we can record is thousands of neurons simultaneously. So what you see here in green are all the neurons, and then in red here are the different adult-generated neurons. Here's an image through the 2-photon microscope that this was collected in collaboration with Attila Lasansi's laboratory at Columbia, and we can see that basically there's this kind of activation, this kind of flickering of light as the cells fire. And I'll play this again just real quick. And so you can see kind of as the cells, it's very, very quiet, but then you get this kind of flickering whenever these cells fire. And we can monitor the activity of these cells whenever the animal's running here. And we can ask, how are they different, the mature cells versus the immature cells in the live brain? And so the first thing that we find, if we just look at the activity of these cells and ask who's more active, what you find is that the newborn adult-generated granule cells are significantly more active than their mature counterparts. So they're basically a population of cells in the dentate gyrus that are more active and basically are more stimulated than their mature counterparts. So then the next question that we want to ask is, do they encode information about the environment differently? So as I mentioned, these animals sit on a treadmill and they basically run across the treadmill and they get a bunch of cues as they run across. And now we can look at the activity of an adult-born granule cell and a mature granule cell and ask how they differ in basically how they encode the environment. As I mentioned in the beginning, these cells show spatial tuning. They tend to fire only in specific places in the environment. And so if you look at a mature granule cell, now what we've done is we've basically made the track circular and the animal runs around in a circle. You can see that most of the calcium transients of the mature granule cell tend to fall in one specific area. So they're basically taking cues from the treadmill belt and kind of falling in one specific area. However, the adult-born granule cells tend to fire multiple places in the environment, indicating that not only are they more active, they basically show less spatial tuning and they encode basically less information about the environment, or they actually may encode different pieces of the environment that are not spatially tuned. And so if we quantify that, this is here. So what we're looking at here is the tuning specificity of adult-born granule cells versus mature granule cells. And what you see is adult-born granule cells are basically less spatially tuned. In addition, here, if we look at this spatial tuning metric, what we find is that the more active an immature granule cell is, the less spatially tuned it is. So here in green, we have all the mature granule cells. So the more active the cell is, the more spatially tuned it is, because the transients just fall within the center of its place field. However, the immature granule cells, the more active the immature granule cells, basically the less spatially tuned it is. So that would indicate that within the dentate gyrus, you're generating these young neurons that are highly, highly active, but actually are very poorly spatially tuned. And so what we're trying to understand is basically why you would generate neurons within your hippocampus that basically do this, that have this kind of noisy activity and promiscuous activity, but very, very high activity. And so this would indicate, so just a brief kind of conclusion to this area, this would indicate that adult-born granule cells modulate context encoding and discrimination in fear memory. Both these populations of cells, mature and adult-born granule cells, actively participate in dentate gyrus population activity during exploratory behavior, and that these adult-born granule cells are more active than mature granule cells and exhibit less tuning specificity. So this would suggest that the role of young adult-born neurons in context encoding actually may derive from their unique properties, so their high activity and their low kind of information that they process, and that this kind of increased activity may actually modulate and influence local circuits very differently, and so they may actually influence output of the hippocampus in a very different way. And so how are we kind of following this up to try to understand what these new neurons are doing? So one thing that we've kind of switched to do in parallel to the two-photon imaging is actually use freely moving imaging techniques. So what we do here is we actually use a miniaturized microscope that can mount on the animal's head, and we can image the population activity of dentate gyrus granule cells through a Grin lens implanted above the dentate gyrus. So here what we have is dentate gyrus granule cells labeled again with the calcium indicator. And so what we can do here is you can see the animal here now is running through an environment in a freely moving situation, and we're actually imaging the large-scale population activity within the dentate gyrus. And so now what we can do is we can actually label adult-generated neurons as the animal is behaving and monitor large-scale population activity. So here what we have is basically the activity of multiple neurons within the dentate gyrus as the animal is behaving. And so what we can do now is go back to kind of this scheme where we can look at fear generalization and ask, how do adult-born neurons actually discriminate between safe and fearful stimuli? And by monitoring the animal in real time. And then in addition, what we've been doing is actually breaking down these encoding contexts into their actual elements. And so one thing that we've been looking at is the encoding of fearful stimuli by dentate gyrus granule cells. So this is Nick Woods, an MD PhD student in my lab, has been looking at the ventral portion of the dentate gyrus and asking basically how these cells encode different odors And what happens when we basically make one of these odors fearful? Can we actually see differences within the population activity when the animal has to learn something fearful? And so here, what I'm just showing here is very preliminary data where we basically image the animal under baseline conditions to 3 different odors that are just very neutral to the animal. And then we pair one odor with a foot shock. And then ask the animal basically how they can discriminate now between the odors at the neural level after one of them has been made fearful. And so here what we're doing is we're actually using a decoder to actually ask the neural activity to tell us which odor that the animal's getting. And the dentate gyrus does very good at this. You can decode odors from the actual dentate gyrus activity because the animal gets significant input from the lateral entorhinal cortex, which processes odor information. And what we find is that before the animal's trained, you get very good odor discrimination. And what's interesting in this preliminary data is that you actually get a decrease in the ability to discriminate between these 2 odors after that's been paired with a fearful stimulus, indicating that there may be some generalization at the neural level in the dentate gyrus after fear learning. And then what we also look at is correlations between neurons, and we ask how important correlations between activity of neurons there is, and there actually is here. So those are kind of the methods that we're actually looking at now to kind of probe this circuit a little more to really kind of get a handle on what these adult-generated neurons are doing to behavior. So now I'm going to switch gears a little bit and switch kind of from the local circuits of the dentate gyrus to kind of long-range circuits of the ventral hippocampus. And so, as I mentioned, the ventral hippocampus sends significant projections to downstream areas that are involved in kind of fear, anxiety, emotional processing, such as the hypothalamus, the amygdala, the medial prefrontal cortex, the nucleus accumbens, the septum, and the BNST. So these are monosynaptic connections from ventral CA1 to these areas. And so what we've been looking at is kind of probing this and understanding how this plays a role in behavior. So we originally got interested in this by just an observation we made a few years ago when I was at Columbia. So here we're doing another optogenetic experiment in an animal that's running in a classic test of anxiety in mice, which is the elevated plus maze. We've implanted fiber optics above the ventral dentate gyrus to acutely manipulate activity in the ventral dentate gyrus. The animal basically likes to stay in the closed arms, and it doesn't like to open to the open arms. Now, if we manipulate the activity specifically in the ventral dentate gyrus, what you see is that the animal basically starts exploring the open arms of the elevated maze. So it almost acts as an acute anxiolytic to the animal. So this is the same thing that you would get if you injected this mouse with a benzodiazepine, for example. And so this is reversible. So when we turn off activity, the animals basically go back into the closed arm of the elevated maze, and they don't keep exploring into the open arms of the maze. And so this was very interesting to us because it showed that actually direct modulation of the hippocampus, and specifically what we showed is in the ventral hippocampus, we can get an acute anxiolytic effect. And so this is shown here within the group of animals, that basically the time that the animal spends in the open arms of the maze is increased when the animal basically, when we modulate the ventral hippocampus. And that this is reversible, so the blue ones come down, and the animal basically goes back into the closed arms of the maze after we turn off activity in the ventral hippocampus. This is true across multiple tests of anxiety. Here I'm just showing it to you in the elevated plus maze. So in addition to our finding, there have been other findings before us and after us that have kind of confirmed this issue. So for example, there's been an old lesion literature which has shown that if you lesion the ventral portion of the hippocampus, this is anxiolytic. So this was shown by Bannerman as well as by the Moser lab has shown this also, that if ventral hippocampal lesions lead to a decrease in anxiety. In addition, Josh Gordon's lab has spent years working on kind of synchrony between the ventral hippocampus and downstream areas in anxiety, and what they've shown is actually the ventral hippocampus and the medial prefrontal cortex tend to synchronize when animals explore anxiogenic environments. And then in addition, work from the work that I just showed you, as well as work from Josh's lab, has shown that if you optogenetically modulate hippocampus as well as its outputs, you can have very direct effects on anxiety-related behavior. So this kind of lays the ventral hippocampus in this kind of area that's involved in kind of modulating the anxiety state of the animal. However, the ventral hippocampus is very heterogeneous, so it sends, as I mentioned, multiple outputs to downstream targets. And so what we've been very interested in is trying to understand basically how anxiety-related information is encoded within different populations of ventral CA1 neurons that project to these downstream areas, and whether it may be differentially encoded to control different kinds of anxiety-related behavior. And so the questions that we've been asking is, how are anxiogenic stimuli represented in the ventral hippocampus, and how these representations differ in different ventral hippocampal output streams? So in order to do this, what we've done is we've actually gone back now and used this technology of miniaturized microscopes to test animals in freely moving anxiety tests and determine how different populations of cells are responding to exploration of anxiogenic environments. And so here what we've done is we've now expressed that calcium indicator in ventral CA1, and now we're imaging large-scale population activity. These are ventral CA1 neurons that are basically firing in response to an animal basically exploring. And you can see here the transients. We can record from 50 to 100 cells simultaneously. And now this allows us to use different genetic tricks to tag cells based on their anatomical projection pattern or their genetic profile. And so the first thing that we did is we basically ran these mice through that elevated plus maze and asked whether or not there's a recruitment of the ventral hippocampus during exploration of anxiogenic environments. And so here what we're looking at is the animal running through the elevated plus maze, and we're looking at activity when the animal explores the open arms of the maze versus the closed arms of the maze. And if we look at the calcium transient rate, so this is the number of transients that are fired by the actual ventral CA1 during exploration, what we see is that you basically have an increase in activity whenever the animal goes into the open arms or the anxiogenic compartment of the maze. In addition, if you look across specific crossings, whenever the animal crosses from the closed arm into the open arm, what you see is you kind of mount this response in ventral CA1. You get an increase in activity. And now if you basically flip it in the other way, when the animal goes from the open arms into the closed arm, this kind of activity comes down. So you get this kind of recruitment of ventral CA1 activity when animals are exploring these different anxiogenic environments. And this does not happen when the animal transitions from the same compartment, say from a closed arm to a closed arm or an open arm to an open arm. So it seems to be specific for when the animals tend to transition into these open arms of the maze. In addition, we asked whether or not this kind of change in activity was related to the actual anxiety state of the animal. So we know within mice, you get basically a distribution of how anxious different mice are, even within a genetically identical population. And so here what we've done is we basically plotted the amount of time the animal spends exploring the open arms as a measure of how anxious the animal is with how much of a response they actually mount in their ventral hippocampus. And so what we find is that the most anxious mice, the mice that spend the least amount of time exploring the open arms, every time they actually do explore the open arms, they have a much greater increase in activity in ventral hippocampus. Whereas the least anxious mice tend to mount a smaller response. So then the next question was, well, is this specific for the elevated plus maze, or could it be in any different task that you get this recruitment of ventral hippocampus? So we tested them in another test of anxiety, which is called the open field test, where the animals tend to avoid the center of an open field compartment. And we asked whether or not the activity was greater when the animals were in the center versus a periphery, and it was. So there's basically an increase in activity when the animals enter into the center of the open field. And then we asked, how about in a test that is not anxiety-provoking, that actually animals want to do? So we test them in a novel object task, where animals really like to explore new objects in a familiar environment. And so here what you see is that the animal basically spends a lot of time exploring the novel object. However, it does not mount this response in ventral hippocampus, indicating that this may be specific for exploration of kind of anxiety-provoking environments. And so the next question that we wanted to ask is we want to use this technology to ask whether the same neurons are recruited across multiple tests of anxiety. So using this technology, we can actually image the same population of cells across multiple different behavioral tests. And so what we ask is basically, if a neuron is selective for open arms in the elevated plus maze, is this neuron also selective for the center of an open field? Or is it selective for kind of the novel object zone? So how do these relate? The hypothesis being that you actually have cells that are encoding anxiety that are not specific to the actual specific task, that are recruited across multiple tests of anxiety. And so indeed this is the case. So I don't need to go into this completely, but basically what we do is we generate a random distribution just to make sure that our overlap is greater than chance. And what you find is that the true overlap of open field center cells versus open arm center cells is greater than what you would expect from chance. So you basically have cells that are recruited in multiple tests of anxiety. Whereas if we compare the novel object test to the elevated plus maze, we see that there's no overlap here. So indicating that you get different populations of cells that are recruited in these different tasks. Okay, and so the next question is this specific for ventral CA1? So then what we did is we implanted lenses above the dorsal portion of CA1 and imaged there and asked whether or not there's a different recruitment of these cells. And what you see is that basically while almost 50% of the cells that we record from in ventral CA1 are arm type, open arm selective kind of anxiety cells, in dorsal CA1, it's basically chance. So basically you have about 30% of the cells are closed arm cells, neutral cells, and open arm cells, indicating you have an enrichment within ventral CA1. Okay, so then the question is, okay, so you have this enrichment of activity in ventral CA1, this increase in activity. Can we basically manipulate the animal's anxiety levels in these tests by actually turning down activity in ventral CA1? So in order to do this, we went back to an optogenetic strategy where we express ARCH now, which is the light-sensitive proton pump that allows us to silence ventral hippocampal neurons in ventral CA1. And we applied kind of a behavioral closed-loop test here, where whenever the animal enters into the open arms, we turn down activity by shining light on ventral CA1 to basically bring down activity in ventral CA1 and ask whether or not open arm inhibition can reduce anxiety in these mice and allow them to explore the open arms more. And so, indeed, this manipulation with ARCH caused animals to increase their exploration of the open arms, indicating that they have a reduction in anxiety. We've tested this, we've controlled for this in multiple tests in different compartments of the maze and shown that this is specific for the open arms of the maze. In addition, it's not that these animals actually like this stimulation, because if we put them in a box where we only give light on one side of the box and ask whether they just tend to approach that side of the box, the animal doesn't really care. They kind of explore both. So it seems to be specific for recruitment of these cells in kind of an anxiety-provoking situation. Okay, so as I mentioned before, ventral hippocampus is very heterogeneous. Everything that we've been doing so far is kind of silencing the entire population of ventral CA1 neurons. And so now what we've wanted to ask is, can we parse out ventral CA1 into its specific outputs and ask whether or not different outputs are actually doing different things in behavior? And so we've started our investigation, at least for these early studies, at the projections of the ventral hippocampus to the amygdala and to the hypothalamus. So we know these are two areas that are involved in kind of learned fear and innate anxiety, and we want to ask whether or not the ventral hippocampus is playing, cells in the ventral hippocampus that project to these two areas are playing different roles in behavior. And so very quickly, if you look to see, this is kind of an anatomical study showing that if we inject an anterograde tracer, which is an AAV into ventral hippocampus and look for the terminal fields, you see really nice terminal fields in the basal nucleus of the amygdala here, as well as in the lateral portion of the hypothalamus here. And these cells do make synapses on postsynaptic cells, because if we do in vitro patch clamp recording of these cells, basically we put channel rhodopsin in ventral hippocampus and see whether or not there's a response in the postsynaptic cell in the amygdala versus the hypothalamus, and you see a nice postsynaptic response that's mediated by glutamate receptors, indicating that these cells are making direct synaptic connections. These ventral CA1 neurons are making direct synaptic connections onto the lateral hypothalamic neurons and the basal medial nucleus of the amygdala neurons. So then the next question that we want to ask is it the same neuron that projects from ventral hippocampus to these two downstream targets? So in order to do that, we inject retrograde tracers now into these downstream areas, and we look upstream at ventral CA1 and ask whether the same neuron in ventral CA1 sends a projection to these two areas. And what it looks like is that they're largely non-overlapping cell populations within ventral hippocampus. So basically you see very few cells that are co-labeled for both. They tend to basically lay in different layers of the ventral CA1, indicating that within ventral CA1 you have two populations of cells that send projections to two separate downstream targets. And so, and this is quantified here, and basically these cells tend to lay in different layers of ventral hippocampus. So the question became is, if we manipulate the activity of these cells, can we have different effects on behavior? So we started with the hypothesis that perhaps the amygdala projecting ventral hippocampal cells are important for context fear learning, because we know that the amygdala is involved in taking contextual representations and pairing them with fearful stimuli, and that perhaps the hypothalamic projectors are important for this innate anxiety-related behavior that we're seeing. And so The first thing that we did then is to control the activity of the ventral hippocampal projectors into the amygdala by implanting fiber optics above the amygdala and now manipulating the activity of ventral hippocampal inputs to the amygdala and testing the animals for their ability to encode or retrieve a contextual fear memory. So it's very similar to the neurogenesis experiment I showed in the beginning, where we can manipulate the activity only during either training or during retrieval of a contextual fear memory and ask whether these mice can learn it. And indeed, if you manipulate the activity from ventral hippocampus to the amygdala, you basically impair the animal's ability to basically learn a contextual fear memory as well as retrieve it, indicating that this pathway is important for the learning of contextual fear. However, if we manipulate this pathway, and I'm not going to show you for the sake of time, if we manipulate this pathway, we get no effects on kind of innate anxiety-related behavior as we measured in the elevated plus maze or the open field or any of these kind of innate anxiety tests. However, if we do the same experiment now, however, implant fiber optics above the lateral hypothalamus, now we can get effects on anxiety-related behavior. So basically, stimulation of these outputs into the lateral hypothalamus causes an increase in anxiety, so the animals don't tend to explore anxiogenic environments as much. And in addition, this kind of stimulation is actually aversive. It causes aversion in animals, so they tend to not want to explore kind of the stimulated side here of a real-time place preference assay. They tend to avoid the stimulation. So it's anxiogenic and it promotes avoidance behavior. However, this stimulation does not impact contextual fear memory. So what we're starting to think is that there's a dissociation at the level of the hippocampus based on where these cells are projecting on whether or not they play a role in kind of innate anxiety-related behavior or more learned fear-type behavior. And so then finally, the last experiment that I'm going to show from this series of studies is what we've wanted to do now is image these ventral hippocampal neurons defined by their output. So because we're using imaging now, we can actually label cells based on their anatomical projection. So here what we do is we inject a Cre recombinase into these downstream targets that labels cells presynaptic neurons. They basically go retrograde, this canine adenovirus, and then inject the Cre-sensitive GCAMP into the ventral hippocampus, allowing us to image cells defined by whether they project to the lateral hypothalamus or the basomedial nucleus of the amygdala. And so this is basically just shown here that we can do that these are cells now labeled by basically where they project, if they project to the amygdala or they project to the lateral hypothalamus, allowing us now to begin to really parse out these different populations of cells within ventral hippocampus defined by their projection output. And so what we find is that now if we go back to basically saying whether or not neurons are recruited by the open arms of the elevated maze and ask what percentage now of these two populations are recruited by the elevated maze, what we find is that a large proportion of these ventral CA1 cells that project to the hypothalamus are selective for the open arms of the maze. So about 80% of the cells we record respond to the open arms of the elevated plus maze, whereas we still do get a significant proportion of these amygdala projecting ones, about 47% do respond. However, optogenetic modulation of these cells didn't really have much effect on the animal's behavior. So we're still trying to figure out what these cells are actually doing. And so what we wanted to ask now is if we now turn down activity specifically now in the ventral CA1 projection to the lateral hypothalamus, whether we can get effects on anxiety-related behavior. So now we use the same technique, except we now silence cells defined by their projection to the lateral hypothalamus and ask whether we can have acute control of anxiety in these animals. And we use the same method now where we silence only when the animal goes into the open arms of the elevated maze. And what we find now, like we did with full ventral hippocampal silencing, we can get an acute anxiolytic effect by now just targeted silencing of ventral CA1 neurons that project to the lateral hypothalamus. And so now what we think we found is now a population of cells in ventral hippocampus that project to the lateral hypothalamus that now can be modulated and can control anxiety levels in mice in these tests of anxiety. Okay, so now to conclude here, what we're now thinking about is that information is routed out of the hippocampus to control specifically different kinds of behaviors. And so we know that now what we've shown is that projection from the hypothalamus can control avoidance behavior, while those projections to the amygdala can control learned fear. And as I mentioned in the beginning, from work from the Gordon lab has shown that projections into the medial prefrontal cortex can control avoidance behavior. We have data now showing that these two projectors to the hypothalamus and the medial prefrontal cortex tend to be two two different populations of cells, and so we're very interested in understanding how these two populations actually encode information differently. And so now to link this up with the original adult neurogenesis work, what we think is that perhaps now these two processes in the hippocampus are actually contributing to anxiety-related behavior in very different ways. So perhaps the adult-generated neurons in the dentate gyrus, their role in the ability to discriminate between safe and fearful stimuli can lead to changes in their ability to actually understand whether or not the animal's in an environment that's fearful or safe, can lead to high discrimination and low anxiety. So the animal can basically discriminate fear from safety. In addition, in ventral hippocampus now we have direct effects on innate anxiety-related behavior to control the animal's anxiety state. So finally, what we're doing now is we're basically now asking how emotionally salient stimuli is encoded in ventral hippocampal neurons. And so here what we're doing is we're using two-photon imaging again to image the ventral hippocampus at high resolution and ask what information is actually encoded. Because now by using this kind of a task, we can actually give the animal specific cues that are innately appetitive or innately aversive and ask what information is actually encoded within this population of neurons. And so what we can do is we can give the animal things they like, such as sucrose. We can give the animals things they don't like, such as air puffs to the snout, and ask how these cells are responding. So this is actually within the same field of view. You can see cells that respond to appetitive things, cells that respond to aversive things. And so what we want to ask is basically how the ventral hippocampus is organized, not only in just encoding anxiety-related information, but actually encoding unconditioned stimuli that are both appetitive and aversive to the animal. And then finally, what we want to do is define cells based on their projection to downstream targets and ask how they respond to different anxiety tests and stimulus responses. But also now what we're doing is trying to develop techniques to basically target not only the downstream target of the cells, but also the cell type that they're actually synapsing on. So this is a collaboration with Vikas Sohal's lab, who is interested in medial prefrontal cortex and the control of anxiety-related behavior, and specifically in one population of inhibitory interneurons, the VIP expressing inhibitory interneurons. And so here what we've done is we've used a rabies-based tracing approach where we can actually label all the presynaptic partners of VIP interneurons in the medial prefrontal cortex, and we can label these cells in the ventral hippocampus that are not only defined by whether they project to the prefrontal cortex, but whether they project to the VIP interneurons or to a different population of cells. And so we want to use these tools now to really kind of get in and dissect how the ventral hippocampus is contributing to anxiety-related behavior and how they form a node within a network that actually of brain regions and cell types that may encode these different kinds of anxiogenic stimuli. So with that, I'd like to conclude and thank the people that have done most of this work. So Jessica Jimenez was a graduate student that works with me at Columbia University in collaboration with Renee Henn to do the ventral hippocampal imaging. Jeremy is doing the ventral hippocampal two-photon imaging. Nick and Victoria are working on the adult neurogenesis project. And then the early adult neurogenesis project was done in collaboration with Attila Zonsi's lab and Nathan Danielson's lab, and as well as kind of my different funding for different parts of this project. Thanks.
I'll start.
Okay.
So the first thing I want to say is extraordinary talk.
Yeah.
It's an incredibly complex topic, particularly for folks who are not spending all their time thinking about ventral hippocampus. And at least I thought it was just incredibly clear and beautiful. But I also just want to say as a general proposition that I think for those of you who don't spend a lot of time looking, so we realize this isn't our normal grand rounds, that this is contemporary neuroscience now, and it's so remarkably different from how things were even 5 years ago, that not just because of the work of Mazen, but in part because of where he's taking the field. The ability to study and dissect circuit-level phenomena in alive, awake, behaving animals is profoundly new, and you can see the kinds of questions, the logical kind of questions that we can now ask and answer deliberately are just things that, honestly, we couldn't think about that just a few years ago. So thank you for that. So now I want to ask these hard questions. Okay, so the first thing is I just want to get your take on what you think you're studying. Yeah. So you can say, well, I'm studying circuit level behavior, and that's incredibly important. But I was listening carefully to sort of how you describe the behavior of the animals as you went through the talk. And so you started talking about fear, then you started talking about anxiety, then you started talking about anticipatory fear, and then you started talking about innate anxiety. And what I'm wondering is one of the big gaps between the work that you're doing and the work that a lot of people in this room are concerned about is sort of how does this relate to our understanding of behavior in humans? And psychiatrists, I think, as a general proposition, might really begin to think about differences between the kinds of things that you're talking about, that anxiety in our patients has certain qualities that are sort of higher order cognitive functionings compared to something like fear, anticipatory fear, or quote-unquote innate anxiety. A mouse knowing not to go into a place where it could be caught and eaten may be really different than someone, being concerned about their next test. And so what I'm wondering is, can you talk about sort of how you think about those terms? Do you believe that they're applicable to our understanding of anxiety, or are you talking about something that may be fundamentally different from what we think about in psychiatry as anxiety behaviors?
Yeah. I mean, so when we develop these kinds of animal models that we use to study these kinds of phenomena, we're basically trying to model behavior. So we basically have a number of tests of validity of whether or not these animal models are valid, whether they have face validity, whether they have predictability, whether they have construct validity for the actual different kinds of behavior. So, for example, one of the main tests that I talk about is the elevated plus maze, which is an avoidance behavior test and a risk assessment test and a threat assessment test, right? So individuals with anxiety disorders tend to avoid kind of social interactions, tend to avoid different kinds of things that avoid exploring their environment in some way. So we know that the rodent situation kind of looks like that, right? So basically that's kind of the closest that we can get to trying to understand that specific feature of whether or not animals tend to kind of explore and kind of the explore versus safety kind of dilemma that the animal has. We're tending to model that. We know that different drugs in rodents like benzodiazepines can change behavior here that also change behavior in humans, and so chronic SSRIs also change behavior in these different tasks. They also change behavior in humans. Also, we tend to think the same neural circuits are important in the rodent and in the human in these kinds of tasks. So we pick tasks that we know that basically are engaging the same neural circuits. So for example, when we test an animal in their desire to explore an environment, we know if they give a chronic SSRI and modulate serotonergic tone in the brain, we can get changes in behavior here that are made that in rodents that kind of are similar to changes in behavior in humans, right? So that's kind of the innate fear side of things. Also, I talked a little bit about kind of learned fear and contextual fear. And that, what you hear when you come to these kinds of talks, these are developed as models of, say, post-traumatic stress, right? So the animal gets basically a fearful stimulus and then is asked basically to distinguish between a similar representation to that original fearful stimulus. The idea being that if it's similar to an individual that develops post-traumatic stress, say, from being attacked in a parking garage, and then they tend to fear all parking garages. That's what we're trying to model here, right? And so we tend to think similar neural circuitry are involved in those two aspects too. So for example, in amygdala is important, the hippocampus is important, and connections between those two areas are important. So what we're trying to do now is basically just develop animal models that kind of look like the human condition and that resemble kind of that the circuits and the treatments that have been somewhat effective in humans actually have behavioral effects in rodents.
Okay.
Yeah.
So I want to push you a little bit unless someone else wants to join in on this conversation.
Yeah.
Okay, so you talk about different kinds of validity for animal models, and I'll reveal my bias. So as a human geneticist, my feeling is that the only construct valid model for human neuropsychiatric disorders would be a gene that was shown to be able to lead to that disorder in humans, and that would give you an idea that there's a molecular link between the thing that leads to something in humans and what you're doing in mouse.
Right.
So what you're talking about predominantly is face validity.
Yeah.
Okay, so, and there may be other ways to think about construct validity, but from my standpoint, and face validity is a great way to begin to explore, but we also know profoundly limited, right?
Yeah.
Like the reason that Big Pharma is getting out of the business of developing new antidepressants is that face validity for mouse behaviors at a superficial level led to zero.
Right.
So you know that SSRIs can fix it, but if you go in the other direction and say, do things that fix this otherwise lead to medications that are going to help humans?
Right.
The answer from Big Pharma largely has been no.
Yeah.
And so then one of the questions that I have is whether or not you think that the reason that those things failed was an insufficient understanding of circuit level phenomena.
Yeah.
And so whether you think that this approach may be able to rescue the idea of face validity for things like therapeutics development.
Yeah, I mean, absolutely. I mean, I think that understanding kind of the circuits that are mediating these different kinds of behavior that may be kind of generalizable across multiple disorders. So avoidance behavior is generalizable. I mean, these are not, we're not testing a specific disease here. We're testing just a behavior. And so we're trying to find a neural circuit that's important for normal behavior, which is adaptive in animals, and ask basically how these circuits are contributing to this behavior. And then if we can find targets, like, so we think ventral CA1 to lateral hypothalamus is an interesting target, and we're doing all the molecular work now to try to see if we can actually specifically target those neurons and see if we can basically control the behaviors we look at here, right?
Great.
Yeah.
Good question.
Thank you.
And I'm entering into it from a triangle. If I understood you correctly, if we had an imaginary basket of the experimental mice you've been using, we would find by putting them into like the open arm test with the elevated pace, that some of them were temperamentally more likely to behave in a way you're calling anxious avoidance. And some were temperamentally, and they were all drawn from a very similar genetic pool of laboratory mice.
Yeah.
So that relates to the genetics issue. They're genetically similar as far as we know. They have what, if we were to jump to the adult anxiety population, more of a personality disposition to fearful behavior and avoidance behavior. What's the difference in those mice?
Yeah, I mean, it's a great question, and I don't have an answer, honestly. So we know that within a genetically inbred population of mice, you do, as I showed you there, you do have a range of behavior that animals basically don't all behave in an identical way. They may respond to the experimenter in a different way. They may basically have, different, as they've been bred in for generations and generations and generations, there may be some drift within the population that may change their actual neural circuits. What's interesting about this is that not only are they behaviorally different, the actual neural signature that we're recording is actually different. And so I think that's a great avenue to look at, is to actually, maybe we can understand the behavior by understanding how these mice are different within the actual specific population. The problem is that these experiments are very difficult to do in large numbers at this point. And so we can look at behavior, and we can look at things like genetics. We can look at kind of gross measures of circuit function, like how is this activated by this specific task. But to get at kind of very large neural circuit recordings, I think takes time, and eventually we'll get there. I mean, that's where the field is headed. But I think it's a great way to kind of get a handle at what are the neural circuits that are mediating the behavior by just looking at individual variability within populations.
So, Marty, one of the things that I didn't mention when I asked that question is that one of the reasons that Mazen may not be doing what we at human geneticists say is a construct-valid model of anxiety or depression or PTSD is that so far gene discovery in those areas in human populations have basically been not terribly useful. In mood disorders recently there was a genome-wide association study that showed a handful of genes now for the first time that sort of meet gold standard statistical thresholds. The problem is that the study needed to be so large to find, and the effect size is so small that it's not clear to people in the field whether or not those are just random noise within a study because they used 200,000 people, and the studies were never designed, so surprisingly overpowered or not. And so this challenge about understanding the humans or mice with regard to their genetic substrate for anxiety, PTSD, and mood, unfortunately, is limited.
Yeah.
Other questions? Yes.
Unfortunately, I think most of us are not familiar enough with the methods to ask you very sophisticated questions. So we ask metaphoric questions that might relate to psychiatry, and it gets at the question you originally asked. It seems that much of what we do in psychiatry is trying to decondition conditioned stimuli. And you have to be impressed that it's hard to do that.
No.
The fearful conditioned responses are so hard to decondition that we spend hours,
Yeah.
and maybe we're not always using the best techniques.
Yeah.
Maybe if we went right to exposure therapy, it would be a little bit more effective, but it's still hard to,
Yeah.
to do that.
Yeah.
Any thoughts at a kind of cellular? I don't know if there's anything you're doing to
Yeah.
notice that.
No, I mean, so I haven't talked about that here, but I mean, we try to model that in mice. I mean, we run animals through extinction as a measure of kind of an exposure therapy type of experiment where we condition the animals to a fearful stimulus and then continue to recondition them, to re-expose them to the stimulus over and over again to see a reduction in the animals freezing. And we try to understand what are the neural circuits that are actually important for that. And actually, there was not my work, but there's actually a recent paper that indicates that ventral hippocampus, which is an area that I've been looking at, projections from ventral hippocampus to the central nucleus of the amygdala are actually important for the renewal of fear, the cue-induced renewal of fear after extinction. So basically, after you extinguish an animal, and you basically show that the animal no longer shows fear to the actual conditioned stimulus, and then you expose them to the original context that the fearful event happened, and you basically play the conditioned stimulus, the animal will freeze again. So that's this process called fear renewal. And it's been shown that the ventral hippocampus is actually important for that process. It's important for basically the renewal of fear after it's already been extinguished. So we're trying to model that and trying to understand the neural circuits that are important for this process of extinction, trying to see if we can accelerate the process of extinction in animals. But yet we don't have tons of evidence right now. I mean, the amygdala seems to be important, specifically circuits in the amygdala. How the hippocampus modulates that still actually remains unknown.
This question, I guess, goes to maybe a specific example of the question of face validity in connection with the adult neurogenesis work, where I was trying to get my brain around the idea that you see this enhancement of adult neurogenesis in the enriched environment, but that your example of what I think we used to call an impoverished environment in these kinds of studies way back when is now being equated with stress. And now it goes to the question of what adult neurogenesis and hippocampus is doing and what it's needed for, and whether in fact this is really a stress dimension or whether it's environmental challenge or complexity question or dimension.
Yeah, so I mean, what I showed was the social isolation experiment, but people have done similar experiments with chronic stress, chronic unpredictable stress in rats and mice, and show that there's a reduction in neurogenesis. So the closest I can come to answering what you're asking comes from rodent studies. And so if you ablate neurogenesis in a rodent, if you get rid of all neurogenesis and you take the animal and you restrain it and you look at whether the animal mounts a glucocorticoid response to an acute stressor, you see a reduction. Basically, it suggests that the output of the hippocampus to the hypothalamic pituitary adrenal axis is affected if you manipulate levels of neurogenesis. And so all I can say with the neurogenesis stuff is that somehow those neurons are doing something to the circuit to manipulate the actual output of the hippocampus and the control of stress circuitry. But how it's actually doing that, we have no idea yet. So I actually don't know how it's actually doing that. We just know that it's modulating the activity in some way.
If you were to manipulate stress independently at the level of enrichment in the environment,
oh, I see. So
ways to do that.
I don't know. Yeah. It's actually a really great experiment. I actually don't.
Would they still mount the?
Great experiment. I don't think anyone's done that. Maybe I'll do that. That's a great idea. We should so kind of mix together enrichment and stress and compare. Yeah. So I think that's a great idea.
Over here.
Yeah.
Two questions. This first is getting at validity again. Could you comment on analogous structures in human hippocampal circuits versus your rodents? And the second question is regarding, I actually forgot it.
Okay, so
the first one was good.
The first question,
the second one we'll come back to you. Go ahead.
Yeah, so the first question is, so there's a really cool study that by this guy named Dominic Bach in I think he's in Austria. So what he found is that if he puts humans in fMRI machines and does virtual reality as the animals walk around and enter into large open environments. The humans, yeah. I think about things in animals, yeah. So he has humans basically walk in a elevated plus maze, very similar, where the human has to walk and has these open areas around them where it's a little bit frightening. They see activation in the anterior hippocampus of these individuals, so indicating that the ventral hippocampus, which is the analogous structure in the rodent, is recruited in a test that's very analogous in humans. Yeah.
And then the second question.
Okay.
How do the adult-born neurons become the mature neurons?
Yeah, I mean,
About that concept.
yeah. So at least that process from a developmental standpoint with all the transcription factors and everything like that has been really worked out very well by people that study these kinds of things. And so basically it's a very similar process as an actual during early development. So it tends to be the same transcription factors, the same molecules are involved in forming a dentate gyrus very early in life as it is in adulthood. So if you basically manipulate a lot of these factors in the adult brain, you get deficits in adult neurogenesis. So it seems to be a recapitulation of an early developmental process.
Here and then here. Sure. I had also two questions.
Yeah.
One is like a pretty specific question. So I was looking back at your slide, or I was remembering there you had a slide in which you're stimulating the amygdala, or sorry, the ventral hippocampus to amygdala projection using a channel rhodopsin.
Mm-hmm.
And I think you got a result that I was sort of like non-expected.
Yeah.
You basically saw a decrease in the amount of potential anxiety
Yeah.
related behavior when you might have hypothesized, according to your model, that the opposite would happen.
Yeah.
That stimulating the projection into the amygdala would have caused more anxiety.
Yeah.
And is that something that
Yeah, so
you expected? Or?
I expected the results shown here for one main reason is that I find that so natural activity patterns in the brain are important, right? And so when you drive activity into a structure, even like the amygdala like that, and you drive this, we're now looking at learned fear. We're looking at the ability of the animal to encode a memory or retrieve a memory. We're basically plugging noise into this circuit, right? And so basically it's analogous to silencing it in my mind. In a lot of these studies, whenever you stimulate in a projection pathway, you're basically just pumping in a bunch of noise, and so you're disrupting any sort of natural pattern of activity. So the fact that the animals couldn't learn the contextual fear memory or retrieve it actually kind of made sense to me when I saw the data, because I was like, oh, of course, because there's no natural patterns of activity here. We're pumping in 20 hertz stimulation into the circuit. Yeah.
But I guess to maybe follow up on that, if you block the endogenous activity, basically using like Arch or Halo,
Yeah. We did that. Yeah, we did that experiment, and you get the exact same effect. Yeah, you get the same result. Yeah.
Yeah, so I did like both anxiety disorders, so I can't help this talk just thinking about like how it was sort of for discovery how it's back in my practice back 10 years from now. And so I'm curious to what your ultimate fantasy is, like if you're hoping that this kind of research will lead towards more targeted set circuits in terms of pharmacotherapy or some other programs where you're thinking that, oh, this could be some sites for deeper stimulation or other neurological efforts more about thinking about ways to prevent targeted neurogenesis.
So I would say all three of those. Those are all great. Yeah, so we think about things in terms of deep brain stimulation targets. With any of these optogenetic studies, we're looking for kind of new targets that we can stimulate to change behavior. As I mentioned, we are excited about doing experiments like that last experiment where we find a specific cell type that projects to a specific area, because that means that we can get a genetic handle on that neuron, because we can actually transcriptionally profile that cell and see what makes that cell different than its neighbors, and see whether we can get a new target that we can just specifically manipulate that population of cells. For the neurogenesis, there are many labs now working on small molecules to increase levels of neurogenesis, with the hope that maybe increasing levels of neurogenesis in a human may be therapeutic. There are other ways that you can increase neurogenesis in humans if humans are analogous to rodents, with exercise and learning, things like that. And so you can use more behavioral method to basically increase neurogenesis to specifically target those individuals that may have deficits that are similar to what we're seeing in rodents, like a discrimination deficit or something like that. So all of those things, would be amazing.
So before we move over there, I just want to say, so it's a fantastic question, and as chair of this department, the question about how we're going to leverage this kind of very basic work to make a difference in the lives of our patients is like clearly a major preoccupation. So one of the things that I just wanted to give you a sense about kind of where the department is in thinking more broadly about that question. Just give you an example. So one of the things that Mazen participated in is about 6 months ago, we brought together a bunch of people from UCSF and Stanford to talk about sort of what was going on with therapeutics development in mood disorders in particular. To ask the question sort of because there's this, particularly from the standpoint of how do you leverage basic science, not what clinical trials need to be done, et cetera. Because there's this massive retreat from therapeutics development in large pharma. And just at a 30,000 foot level, one of the things that's so exciting, I think, about our environment here is that we brought together Vikas and Eddie Chang, who are working directly in human brain in order to understand circuit level function. Andy Crystal, who's working on EEG, the overlap of sleep, human physiology, and mood. Mazen, working on elaborating the circuitry in mice. Carl Dieseroff, who just is doing everything, including understanding every cell in the brain of zebrafish and how that works. Mark Van Zastor, who's at a cellular level, beautiful cell biologist, trying to understand molecular mechanisms. Who else?
Adam Ghazali,
who's thinking about can we manipulate the same circuits using non-invasive technologies. Amit Etkin at Stanford, who's trying to kind of do a similar thing. If you can identify a circuit that's relevant, can use TMS or some other, cognitive behavioral approaches to target specific circuits. So what you can begin to see, I think, in our environment here is that there are multiple levels of analysis that are going on simultaneously, and at least my answer to your question would be that it's the way that this fits into a picture that builds from a molecular and cellular to a circuit level to an understanding of human behavior and human circuitry. Exactly how we make those links is the multi-zillion-dollar question, but I think we've got a group of people here in a whole variety of areas, and I think it's one of the things that makes right now UCSF remarkable in the depth and breadth of the neuroscience community and its intersection with clinical interest, not just in neurology but psychiatry. Yes.
Picking up on that, do you see a way of linking this type of research to the effects of psychotherapy, not just cognitive behavioral, but psychodynamic therapy?
Wow, you want to make it really hard? I'll leave that to
Yeah, I mean,
I know that's not your field.
right, yeah.
Is there a
Yeah, I mean, I think that it would have to be at the level of the behavior, so being able to model that kind of thing in a rodent. If we can model that kind of thing in a rodent, either with like extinct...
Put the rodent on a couch.
Yeah, right, put the rodent on a couch, or like...
We're building
yeah.
couches.
Yeah.
Department, there's a lot of donor money for that.
But I think that if we can develop... I mean, I think it goes back to Matt's first question about validity. If we can build rodent models of these different kinds of behavior and these different kinds of therapies, we have the tools now to go and understand those circuits, right? And to go in and like learn about how these circuits are contributing to the behavior and how we can manipulate the behavior by manipulating the circuits. But I think it really comes down to having good animal models. And so that's why, these animal models that we're using have been around for many, many years. These are the models that have been used to test many drugs for many years. And so that's why we started with them. Because what we're trying to do is understand we're using these brand new technologies to understand how circuits in the brain work, but we need a tractable model, tractable behavior that we can use to understand these circuits. And so
I would take a swing at it. You want the chair swing? So the chair swing at that question is that it's the most difficult question, but I think that there is a path forward to begin to understand it, but it needs what Mazen is talking about in terms of building up a semi-reductionist way of thinking about understanding circuit level behavior, what's important in animals, linking that to humans, and then being able to begin to leverage, sort of modern technology to understand potentially what, modern imaging technology, which unfortunately is limited in spatial and temporal resolution, but there still may be a way. And Marty's question about, are we really talking about temperament, right? And is that really something fundamentally different? Maybe a profoundly important question at the end of this process in order to help the most people. What's the intersection between sort of axis 1 psychiatric disorders and underlying temperament? How can we understand it? But without these kind of semi-reductionist building blocks, I don't see any way to get at that question. We're in the back, sorry.
So you're saying, these experiments are hard to do and hard to do in large numbers, and we're talking about relationships and context and how to observe that. So how often, I'm just curious, I used to do this work like 10 years ago, but how often do you do like a quality assurance of technicians that are handling the mic the same way, the context outside of the box? of interactions with another agent is the same across, the different days of the week. Is that something that could be also affecting anxiety, mood, model?
Yeah, I mean, it's a fair point, and it's something that we control for in the laboratory as much as we can, right? We test our animals on the same time of day. We basically run animals all the exact same way, and we try everything that we can to control for those. But I mean, it's a fair point, and it comes with, there's always going to be some experimental, some variables there that are not as well controlled, but we try to control them as much as we can.
Yeah. Those are variables that are actually really important for anxiety.
Yeah. And so we basically try to control in all the mice that are tested to do it the same way across mice. But yes, perhaps across labs, we try to say exactly how we do the experiments, try to replicate exactly how other labs do their experiments in an attempt so that we are doing our experiments in the same way. But there are always going to be these kinds of things that, these issues that come up with this kind of work.
Yeah, I just wanted to make a comment. I actually work in Eddie Chang's lab. He's a neurosurgeon here at UCSF who, in the lab, we do a lot of recordings actually for people's, the human hippocampus and the human amygdala. And a lot of the correlates that we're finding of mood and anxiety are exactly the types of circuits that Mazen's studying right now. So we're getting to the point where we feel like we can actually almost decode somebody's mood based upon the activity of these circuits. And so there is, I think, a certain type of validity, functional validity that we're trying to develop. And I think the types of experiments that you're doing are sort of really nailing down the details because we can't really do the same type of circuit dissections that you can do.
And I'm familiar with that work. It's gorgeous work. And what the hope is that perhaps we can find different circuits that you guys will have access to in your individuals and basically run them through these same kinds of tests and see, whether there's connections there. Yeah.
Great. Thank you all. Thank you.